Assalamu alaikum everyone. First of all, I am very thankful to International Conference on Applied Zoology for providing such grateful platform to share my work on entomology. I am Muhammad Shahzad, MSc graduated from Department of Zoology, University of Education, Lower Mall Campus, Lahore, and my topic of presentation is biopesticidal efficiency of crude venom Hotentota tamulus against aphids. I acknowledge efforts and contribution of Dr. Muhammad Arshad and Dr. Shahzad from Department of Zoology, University of Education, Lower Mall Campus, Lahore. Let's start with introduction. 10,000 species of insects are considered as a pest of crops. Annually, 14% of total worldwide crop production and 20% stored food grains are destroyed by this large group of pests. The estimated loss each year is about $10 billion. Pesticides have adverse effect on human beings, beneficial insects, pollinators, bird, aquatic, and marine life, so that about 169 insecticides were banned from January 2005 to December 2009, and only 9 new insecticides were registered during the same period. Use of biological product to control pests is an effective method as compared to use of synthetic insecticide. Background: the most effective and the dominant method of controlling insect is use of chemical insecticide. Investment of $1 on the crop in the form of insecticide to protect them from pests returns $4 in the form of increase of crop yield. Uncontrolled use of insecticide creates many problems such as harmful effect on the organisms that are beneficial for the environment. They are also responsible for the development of resistance in the insect against insecticide. They accumulate in the food materials and ultimately cause severe disease in humans like cancer, kidney failure, and genetic disorder. Biopesticides do not cause any problem to the environment because they are eco-friendly. Biopesticides are derived from the natural product. In this case, scorpion venom is a mixture of neurotoxin. Most of these peptide chains of amino acids, these amino acids interfere with the membrane channels that transport sodium, potassium, calcium, and chloride ions. There also has a pharmaceutical uses such as anti-cancer, antibacterial, antifungal, antiviral, antiparasitic, immunosuppressive drugs. Objective of my study: according to the most recent United Nations 2012 Vision of World Population Prospect, population of world will increase from 7 billion to 9 billion in next 40 years, but the loss of the crop due to insect is creating a great problem for us to feed world increasing population. For competing with this problem, the yield of crop production must be increased about 40%. This cannot be done without applying pest crop management system. The present study is planned to evaluate potency of crude scorpion venom to control aphid, which is most destructive pest throughout the world. Methodology: materials, distilled water, fresh HCl, which has buffering capacity, scorpion venom, battery plate, macro pipette, trips, tweezer, electrical stimulator, saline solution, and UV lamp. Scorpion collection and maintenance: scorpions were collected from mud houses under bricks, stones, sandy areas of the district Miawali. The apparatus used for this purpose was battery operated portable UV lamp, captured by using 12-inch forceps and placed into jar with proper aeration. Pest collection: aphids were collected from the mustard field of King Morkosur. The insects were collected by hand picking method. The insects were kept in the laboratory at room temperature. Venom extraction from the scorpion: extraction of venom from the collected scorpions was done by applying electrical stimulation method of Osgood and Philosophy 2004. The suitable current intensity was 25 volt for 3 to 5 seconds for the extraction of venom. Venom was diluted with distilled water and preserved in Eppendorf tube. Insecticidal potential of venom against aphid: 40 aphids were removed from the jar and divided into 2 groups, control group and experimental group. Control group contained 10,000 aphids. Experimental group was further subdivided into 3 subgroups, and each subgroup contained 10 aphids. Each subgroup was treated with different concentration dose of scorpion venom such as 0.5 macroliter, 1 macroliter, and 1.5 macroliter respectively. Following the oral method, venom was mixed with the food of aphid and spread on the battery plate. After every 6 hours, mortality of individual was observed in each subgroup. Statistical analysis: One-way ANOVA test was applied to calculate the mortality in different groups. Statistical package for social sciences was used for this purpose. Result: percentage mortality after 6 hours. It has been represented in graph. Percentage mortality with 0.5 microliter was observed 16.67, and percentage mortality with 1 microliter was observed 23.33, and percentage mortality with 1.5 microliter was observed 46.67. Similarly, percentage mortality after 12 hours was represented in the graph. Percentage mortality with 0.5 microliter was observed 33.33, and percentage mortality with 1 microliter was observed 63.33, and percentage mortality with 1.5 microliter was observed 93.33. Percentage mortality after 18 hours. Percentage mortality with 0.5 microliter was observed 73.33, and percentage mortality with 1 microliter and percentage mortality was 1.5 microliter was 100. Table shows the comparison of insecticidal effect of scorpion venom against aphids. In the table, the upper right corner, the time are represented and concentrations are on the left side. First column represents the control group, in which there is no mortality was observed, but when the concentration was increased 0.5, 1 microliter to 1.5 microliter, there was significant increase in the mortality. Table 2 calculated LT50 and LT95 for aphid exposed to different dose of crude venom. LT50 and LT95 in the table showed that as we increase the concentration, LT50 and LT95 decrease. Conclusion: venom of scorpion is rich source of peptides which are toxic for insects. These peptides can be used as a biopesticide and can replace chemical insecticide. Our next aim is to isolate the peptides which are actual toxin for insects by fraction method. If we are able in doing so, then that information will provide us the raw material for synthesis of selective biopesticide at industrial level. Thank you for your attention. Allah Hafiz.